for this nice presentation, introduction. So yeah, as you can see, my talk today will be about data-driven design strategies that we are developing in our lab for biomanufacturing. And I would like to give you first an outline of my talk. So basically, there will be two parts. The first part will be more like a motivating ideas about the design, build, test, and learn cycle and the need of AI and automation. And secondly, I will zoom on biosensors, which is what we are now developing in our lab and how they can be related to all of this approach, including different tools for biosensor design. So I guess that by now everybody here agrees that the vision for a sustainable bioeconomy is something that is an important topic nowadays. That vision, well, there are some estimates like in this report that tell us that perhaps 60% of materials could be produced biologically, like in the consumer market, by just starting from raw feedstocks and through bioindustrial manufacturing. Okay, that view has been there for a long time, but the point is that at the moment still there are, I believe, more questions than answers, because is that really technologically feasible or environmentally sustainable, economically viable, ethically responsible, socially acceptable? So there are many questions. Probably I won't be able today to answer all of them, but I will focus mostly on the technologies that we are developing, technologies that are transformative, and they are bringing us solutions that probably can address all of these questions here. And they are basically SynBio, automation and the DBTL cycle, and AI. What happens is that in the last years, there has been a lot of acceleration on the automation of the design, build, test, and learn cycle of synthetic biology by introducing a large number of robotic platforms and automated systems. And machine learning has been always present as an overarching technology for this, because at the end, you can apply it not just for learning the rules of design, but also for instance, experimental planning, smart sampling, or model inference. So this is normal nowadays to see these labs like here, this foundry or many other biofoundries, where there are almost no operators. No, everything is just based on the robotic platforms. Then what we try to understand, and this was a work that was mainly performed at the Manchester Biofoundry, was to see what are the typical steps that you will see in the DBTL cycle, so that then you can work as is SynBio were a manufacturing technology, because we can automate every step here. So typically you start with the design by looking at the pathways, the enzymes, the DNA parts, the design of experiments, and then you move into the build where they are assembled, and then after to the test part where the cultures are grown, the strains, and they are tested, for instance, using mass spec or other omics technologies. And all of this data is fed back into the learn part for deciding, for instance, if you want to switch to another pathway or to alternate enzymes. But at the end of the day, I think that nowadays the vision that we have is that actually AI can be present everywhere here. Not necessarily has to be only something that is applied at the end of the cycle by using machine learning, but also can be done, for instance, for looking for pathways, looking for enzymes, or optimizing the designs. I could provide you with many examples of this, of how this is now being applied, but just to focus on one case that is at the beginning of this cycle when we are at the design. So there has been a lot of proposals coming from different groups about basically decomposing any molecule or any reaction into their fundamental graphs or fingerprints so that this can be encoded and used to train a neural network or some other type of machine learning algorithm. And in that way, we can, for instance, look for bioretrosynthetic pathways, or alternative ways of producing a similar compound by using the right selection of the enzymes. So here we are using fingerprints, we are using different representations, but still there are several challenges. First of all, because usually these databases, they suffer from a lack of well-curated data. And also this is a problem that is highly combinatorial, okay? So there's a lot of complexity when you start to try to explore all the metabolic space. That's why what we have been doing in the last years has been to provide our own approach, which is quite systematic. And basically, that what performs is to start with a knowledge database, like this graph database that is called Biochem4J. And from there, you can extract the reaction rules that are representing every possible biochemical transformation at different levels of detail, different levels of specificity. This is a powerful generative system because it allows you to go from the known metabolic space into predicting other compounds that potentially could be present there because they fit into the same basic reaction rule that you have here. And there's a lot of body of evidence that the representations are quite close to what actually is happening in the enzymes, that they have this capability of what usually we call promiscuity of accepting different substrates or producing different products. So this is something that we have been investigating by looking at the mass spec spectra to see if actually these peaks that exist there and nobody have been able to match to any given compound, if there is a good candidate coming from our own predictions based on the extended metabolic space. We did this prospective study that was very right at the beginning, and it was very interesting to see that there were some growing evidences providing us this type of approach actually was going to be useful. For instance, just to mention one example here, this is the extended metabolic space, the gray line compared with the orange, which is the spectra that you see typically in a sample. So if you compare this with the distribution of the peaks that you have in the metabolic database, is obvious that we are missing a lot of peaks that potentially are present. And this is what we are seeing here. This last point here is because we limit everything just to 500 Daltons, but you can make it for larger metabolites as well. And then another important thing that we also observe here is that when you are looking at the reaction space, you can see things that are pretty much related to evolution in a similar fashion as what you see when you are looking at homologous sequences. So there is like a parallel between the sequence annotations and the metabolic reactions. And we did several studies based on this across the tree of life so that we show that you could even reconstruct the tree of life based on the chemical space rather, or biochemical space, if you want to call it this way, rather than the sequence space. And this is because what we are observing here is something that is coming from this capacity of enzymes of having a promiscuity, which allows them to evolve and to at some point to accept novel substrates, and in that way create a new alternative pathways. Recently, actually, I was I wanted to bring this recent paper that we have just published, where, well, it's accepted. I think it's still in press, where what we are here looking is at the possibility, or even using the reaction space, the chemical space, to go even uh, to earlier stages in evolution, at the beginning of life, at the origins of life. Because as you probably have heard, there are these proto-metabolic networks that still are not very well characterized, but there are some hypotheses about how they evolve into the clusters of sequences. And we believe that in between, what we have are these clusters of reactions that provide the link between this proto-metabolic network and the current extant metabolic networks. So this is an interesting hypothesis that we expect to study in more detail in the next years. And as I said, this is then about the biochemical space. But when you look at the sequence space, again, if you want to find the right combination of enzymes for producing some molecule of interest, you need to find good candidates. Here, for instance, we have been working for a long time with the flavonoids pathway. And basically, we end up with this tool that is called Cell Enzyme. That is a tool that is specialized on providing the best candidate sequences for any given reaction rule. And that's a small difference from what was earlier performed by other groups, because here your entry point is not the sequence, is not your target molecule, but is the reaction. The reaction is what actually determines the best candidate sequences. And our ambition here is to be able to use this to select pathways that not only can work for producing a target molecule, but also for biosensing, for regulating, or for biodegradation. So there are a full catalog of genetic circuits that can be built based on having a tool that allows you to find the right combination of enzymes for the chemical transformations that in principle are your target. So more specifically, what we did was to test this with several proof of concepts, basically by applying the DBTL cycle. In this case, for instance, it was twice, okay, for the flavonoids pathway. And in every case, just a small selection of possible combination of DNA parts were selected based on the DOE, on a design of experiments approach. And what we found is that just by going through 2 iterations was good enough for finding already high producer strains. So this is an approach that, well, is at the end is just to use the optimal design of experiments, a technique that allows you to reduce substantially the number of experimental runs that you need to perform, and can be adapted to the resources that you have in the lab. So basically, by by selecting different promoters or different plasmids and looking at the combinations. So yeah, here you can see just this is the zoom of this existing library. So as you can imagine, existing library is just a small sample of the full set of possible combinations that were possible here. So that means that the approach is nice. It works quite well. I think when this was introduced in 2018, I think it was quite well received by the community, because it's giving you clues about how to work with this type of complex pathways. Here, you will see an example that is more demanding than the previous one, because here, what we wanted to do is to produce all of these molecules that you see here in blue, and also, well, the green and the orange. So everything here is not native to E. coli. The host here was E. coli. And the idea was to perform in a parallelized way every possible combination of pathways here, so that you can try in an accelerated and rapid prototyping way the different combinations. And this we call this a stress test or pressure test for E. coli. And actually was quite successful in some cases, like for instance, for mandelic acid, because we focus on molecules that are monomers for different types of materials. And in this case of mandelic acid, we arrive at titers that were already above 1 gram per liter, like here, which is already levels that are interesting if you want now to move this into scaling up. You are already probably at the point that the strains are already optimized for going into the phase of scale up. And this was done only in 3 months, in 90 days. So you see here, this is a chronogram, like in a factory, when you see every project here at which stage was during the full time frame of the project. So working in that way, so well organized, help us to be able to successfully produce many different compounds. But then still there are challenges, because I mentioned that we try a few combinations of, for instance, of promoters or ribosome binding sites. But if you want to be more precise, no, in your design, then what you can do, and this is what was done here in this example for producing a limonene, was to actually build a full combinatorial library of RBS that could be generated, for instance, with the RBS calculator, and then use them to train a neural network by encoding all the different sequences that we observe and relate them to the levels. This is the fold change. So that is the level of production that you were obtaining for each individual combination of ribosome binding sites in these two genes in this module of the pathway. So the approach, it was quite successful. You can see here the how well there was a correlates the production with the predictions for every point that we tried in the lab. And even we were able to identify those strains or those combinations with the highest titers. So so that I believe that this what is showing us is that sometimes when you are in a very focused problem like this one, it is more powerful to train some predictive system that will learn the context information related to this particular problem, rather than using a mechanistic model like the one in the RBS calculator, because this is not having taken into account all of this information about the context. Another example very similar here was with the promoters. again, we build a library that can select the right promoter for your application. In this case, basically, the idea was to go from an inducible promoter to a constitutive promoter, taking into account the information about the context, which means the vector, no, the plasmid, the type of promoter, the type of strain, and the resistance cassette, for instance, that we were using, the media. All of this information was just fed into the machine learning system. So, and then what we are now is saying, so that we are at the moment that we can design this type of precise types of control and regulation. So then why we should not map the full space so that we look not only to what can be produced, like here this molecule, but also what can be observed by using biosensors, what can be reached, what can be detected. So to try to have a full map of all of these different systems, because then one active field of our research now is to introduce these biosensors to regulate internally the pathway, so that when we change it to the strain into another conditions, for instance, we move it from the lab to the fermenter, still the pathway isn't robust and is giving you a response as you were trying to obtain. So this is something that our PhD student, Jose Eduardo, is working on this. But something that we realize is that in order to work in with this theoretical approach, it is necessary to understand very well the dynamics of the biosensors. And this is something that we just, the paper has just been almost accepted. So I guess that in a couple of weeks will be out there. And what we have done is to train a machine learning algorithm that learns the mechanistic parameters that are describing the dynamics of the biosensor at 4 different context conditions like the ones that you see here. But this is a predictive system, obviously, because it's a machine learning algorithm. So then later you can use it to select other alternative biosensors. Okay, so just to give you an overview of other things going on in the lab. So because we have this interest in working with biosensors, one of the solutions that we have been trying to see if it was giving us a good results was by modeling the biosensor, the transcription factors, because these are basically transcription factors, using 3D modeling, also by training generative AI. And this is where we are now. We are testing now different types of LLM systems to see if that can be used to generate, for instance, the DNA bindings, or especially the ligand binding domain of the biosensor. So this paper that just came out last year actually is Dead Space is the one that provides the full map of everything can be produced and can be detected by using a catalog of around 2,000 biosensors that at the end can be connected almost through almost 1 million of different pathways are possible to connect the targets with the biosensors that at the end can perform dynamic regulation or screening. This is also integrated into a work that is now done by in collaboration with many other institutions. And basically what we try to do here is to have all of these tools as a full pipeline or workflow where you can integrate and exchange information from one step to the other. And in order to do that, we have been developing it on the Galaxy platform that probably you already know because it's widely used in bioinformatics. So what's where we are moving now? So what we are trying to do is to see if we can work more at the level of multi-objective problems, because if at the end what we want is to have pathways that are optimally efficient when we are in large volumes, then necessarily we need to take into account everything that can have some impact on these results, and try, for instance, approaches like Pareto optimality to look for those cases that will provide a best compromise between production and metabolic burden. But still, several challenges are here, because we have noise effects. For instance, we have metabolic burden, we have context effects, as I mentioned before, and all of this is bringing a model dynamics, which are not easy to, for instance, represent by using mechanistic models. Sometimes you have to combine both mechanistic modeling and also data-driven approaches. For instance, recently we were looking at different responses from the reporters in the biosensors, not just looking at the time response, but also using a cytometer to see the population distribution of these fluorescent signals that might need more data-driven approaches rather than a pure mechanistic model. This work was done by Mara Pisani, who was a visiting PhD candidate, and there are other works from other students who have been visiting our lab. For instance, Alexandra, who developed another biosensor for terephthalic acid, or Hugo and Xu, who are working on developing a biosensor for cinnamic acid. So if you think on these biosensors, one interesting problem here that is more a theoretical problem, but I believe that is giving, it might give us new ideas for optimizing the circuits, is that just think on the flavonoid pathway. You can find here across this pathway at least 5 different transcription factors that are responsive to the metabolites that are intermediates of this pathway. So that means if we use the models that we have now for understanding the dynamics of the different biosensors, then what will be interesting is to address problems that are highly combinatorial, are more like mixed integer nonlinear programming problems, where what we would like to do is to find the optimal topologies that will use the biosensors for regulating those genes that are, for instance, are bottlenecks or might have a larger impact in the control of the pathway. So here, this is a collaboration with a group of Irene Otero, and also there is Hector, who is a PhD student. Yeah, these ideas, we already presented this in metabolic engineering in Singapore a couple of years ago, but this year we are now going to bring new results to metabolic engineering 16 in Copenhagen. And well, and with that, I just want to summarize that, as you have seen, we have many solutions that can be used based on machine learning, AI for the DBTL cycle. Still, we have challenges like the lack of well-annotated data, or that not always the models are shared by the community. But we think that as we move into more data-driven approaches, then I think that the best strategy is to combine mechanistic knowledge with machine learning models, like what is called scientific machine learning. Also, to improve transferability and generalization, and focus on optimization and design space exploration. And I just want to conclude saying that we have been in the last couple of years developing this initiative, which is the Valencia Biofoundry here in Spain, where that we have the support from the Global Biofoundry Alliance. And this initiative is, there are many different groups and also technological centers and companies who are involved in this initiative. And we have capabilities like strain engineering, but also plant biofactory and scaling up. So we hope that this biofoundry, which is probably the first one in Southern Europe, will provide these common services for the DBTL cycle to the community. Well, just to thank the different agencies that are supporting our research and all the team and collaborators that we have. And thank you very much for your attention. Now I'm happy to answer any question that you have.
Well, thank you very much for that very informative talk. And yeah, we are opening if anyone has any questions. You can ask.
Yeah, you can just raise your hand or type it in the chat box.
In the meantime, I was wondering for the metabolic space that you're talking about, you usually learn from this database, right? From the database that you mentioned. And
Yeah.
is it also possible to eventually also add constraints from previous knowledge? Like, imagine you are a biofoundry and you are working with a species that cannot do acidic media, whatever. Could you potentially add these constraints to the exploration of space?
Yeah, right. Yeah, this is perfectly possible. I can remember different approaches from the community where one important contribution there was to integrate the wealth of information that they already have about which pathways, for instance, were more efficient, or which sequences. You can do that in a very simple way, like in this type of automated recommendation systems, or you could do it by trying to bring some sort of machine learning system that at the end will predict how likely is that this pathway will be better than another one. But the main problem here is that, well, I can remember some initiatives, like for instance, there was one that was called the laser database, where they tried to bring together many information from the literature, from different groups. And at the end, I think that the main challenge there was that not always these results were comparable, because they were coming from different growth media, for instance, or even different equipment. So I know that, for instance, the IGN community has made a lot of efforts in trying to standardize more the data, but the basic thing is this, and then you're right that when you are in an automated biofoundry where you can generate lots of data, probably is the best scenario in order to develop this type of automated systems.
Very interesting. In the meantime, I also have another question. In the context-dependent biosensor dynamics modeling, paper that you mentioned.
Mhm.
You
Yeah,
mentioned that you use machine learning and machine learning, yeah, a machine learning algorithm. Do you know, could you specify a little bit more what machine learning you use there to infer these model parameters?
yeah, we are. Well, we try different approaches, but the one that we are using now is based on deep learning.
Oh, deep learning.
Yeah,
Okay,
be,
nice.
because we have a technique that performs data augmentation,
Oh,
so that we can train then. Because usually, I mean, the main challenge with a deep learning system is that perhaps you don't have enough data. And here, where we were at the limit, I think, of what could be the. But then we have been developing some strategies for data augmentation, and I think well, it's working quite well.
is that a augmentation something like using the RBS calculator? I mean, information data points and another algorithm to increase the amount of inputs?
Yeah, well, no, actually, in this case, it's more because here the key point is that we are combining a mechanistic model, which is
Hmm.
underlying everything, and then a machine learning algorithm that what tries is to estimate the parameters of the mechanistic model depending on the context. So that means that the first thing that we do is to calibrate those models based on the mechanistic model. And there is where there are some techniques that allow you to generate different, it's like different plausible scenarios explaining these responses that you are observing. So there's no one single solution, and that is quite uh, useful in order to train the deep learning system, because you are telling the system different scenarios that all of them fit into the responses that are being measured.
Thank you. Very interesting.
And I have a question about the ribosome binding site design. So I'm
Mhm.
a little bit wondering how like the machine learning prediction results correlate with the like the RBS calculator results, because this seems interesting to me. And also I'm wondering because the machine learning prediction result is like highly limited by like the sequence coverage you tested for that RBS sequence. So how are you going to design the sequences to train the model?
Mhm. Yeah, well, in this case, I think that there were two, I mean, two main points that were the main reason why we did that. The first thing is, as you mentioned, we were having problem with the RBS calculator because uh, the results were not as expected in terms of RBS strength. I mean, we were able to build a highly diverse library. This is not a problem. But then there was no clue about why some were showing higher strength than the others. I guess that means that depends on which version of the RBS calculator you are using, but at least the one that we were using, we were not happy for this case, except in terms of diversity, because if you look here, and this is in order to answer your second question, actually the design space is because we are just exchanging the RBS for two short regions, okay, in the here in the plasmid. So that means that we don't have so many combinations. I think what you see here, this full list, is whatever could be possible. Then I don't know if you can see it, but sorry, but the ones that are in red are the ones that we measure, okay, and the ones that are in black some of them we tested for validation of the predictor.
Thank you.
Okay.
And any more question from other people here? It's like you can just raise your hand or type in.
Well, if not, you can always email Dr. Pablo Carbonell, and thank you so much for the talk. That was quite impressive, the amount of things that you're doing.
Well, thank you for the invitation also.
Of course, I do have other questions, but probably I'm going to ask you over the email.
Yeah, feel free to send me an email, yeah, with your questions. No
Thank you so
problem.
much. I will, thank you. Thank you
Thank
for
you.
coming.
Okay, bye-bye.